Good afternoon to one and all who joined today with us for this lovely presentation. Respected chief guest, my faculty colleagues, Dr. Shraddha Fish, fellow research enthusiasts, and all the dear participants. I, Dr. Priyal Soni, assistant professor on the behalf of MIT Institute of Pharmacy, AUP Lucknow campus, welcome you all to the online presentation competition in an international conference cum workshop AAE 2023. The theme of our conference is Alternative to Animal Experiments Exploring Imminent Possibilities in Preclinical Drug Discovery Using Computational Tools. And we are fortunate to have among us Dr. Vinod Tiwari. I extend my warm welcome to you. With this note, I would like to introduce our judge to all of you, Dr. Vinod. Dr. Vinod is currently working as an associate professor in IIT PHU since 2018, and his research interest is in neurosciences, specifically in pain management. And Sir has completed more than 6 PhDs under him, and he is having total experience of more than 16 years. He has published many research papers, and the number is 89 publications, and he has a citation of 4416. He has presented many papers in national and international conferences. He is also invited as a person to give many talks. If we talk about his awards and achievements, I want to submit out he as awarded as an outstanding performer in the teaching and research from modern approaches of research in basic and applied sciences. He's also awarded as a Young Investigator Scientist Award by International Brain Research Organization. He is also looking after many administrative responsibilities in various organizations where he had worked previously and currently in IIT BHU. I welcome you, sir. With this note, I want to begin our oral presentation with a quotation that the research that takes place in pharmacy enhances the molecules to become medicine. Data drives the discovery and innovation heals the world. The research we will delve into has the power to impact and only the pharmaceutical industry, but also the lives of the patients worldwide. Over the next few minutes, we'll navigate through the intricate findings, implications, and exciting prospects of the research. So let us embark on this journey of discovery into the world of research.
Thank you for the kind introduction, madam. And I'm looking forward to the participating in this important event. Thank you.
Okay, I would like to call our participants. Our participants have joined already, and in few minutes I will announce our participant. So our first participant is Vikas Chaudhary. I will like to.
Yes, ma'am. Vikas Chaudhary, sir.
Okay, are you available?
Yes, sir.
Dr. Vikas, are you ready with your presentation?
Yes, sir. Ready.
Okay, I would like to inform you that our presentation is just for 5 minutes. So please do not exceed the time limit. All the best.
Okay. Very good afternoon. My screen.
Yes, sir.
Sir, please try now, sir.
Very good afternoon. Today my presentation on oral on topic molecular docking, a new approach for drug research. What is molecular docking? Molecular docking can be defined as bonding of a small molecule called ligand on the
Share your screen. Your screen is not visible to us. PPT. Kindly reshare it. Sir, it is visible now. You may begin now, sir.
Okay, ma'am. My presentation topic on molecular docking, a new approach for drug research. What is molecular docking? Molecular docking can be defined as a binding of a small molecule called ligand onto a specific site in a larger molecule. Docking is the computational determination of binding affinity between molecules, protein structure, and ligand. Molecular docking of 1,5-benzothiazepine using Swiss dock. By the literature survey, we found that the various substitution at 3 and 5 position of 1,5-benzothiazepine zinc yield a compound of clinical interest in our study. Yes, ma'am.
Your screen is still not visible. Please try to reshare it. The presentation mode is not visible. It is visible right now. Yes, sir.
This is visible, ma'am. Hello.
Yes, sir. It's visible.
Okay, molecular docking of 1,5-benzothiazepine using Swiss dock. By the literature survey, we found that the various substitution at 3 and 5 position of 1,5-benzothiazepine.
Sorry, sir. Sir, your screen is black.
Sir, I think you should not do it in a presentation mode. It's
Ma'am, presentation mode जो है.
yeah, sir, your presentation mode is lagging. I think you're facing some internet issue. Please keep it without presentation mode, sir.
Ma'am, slide is visible.
Yes, sir, it's visible right now.
Okay. All computational study were performed using Swiss dock. The 3D structure of following PDB ID were required from protein data bank. The docking score results show very negative score corresponding to a strong bonding and less negative or even positive score corresponding to weak or non-existing in bonding. This slide shows the molecular docking score of the compound under the scheme first, in which 3 substituted CYP bases was selected, in which 25 compounds are selected for molecular docking. Out of 25, only first one A1 to A10 compounds show the good docking score. This slide shows the under the scheme second, in which molecular docking we perform under the Swiss dock, in which the 50 compound were selected for docking study. Out of 50, only first 20 compound B1 to B20 are show the good docking score as comparison to a standard drug phenytoin. And this slide show the scheme third, in which 5 substituted bases were selected, in which out of 50 compound, only first 20 compounds show the good docking score as comparison to a standard drug phenytoin. In result and discussion and conclusion, it was found that the compound A 1 to 25, B 1 to 50, and C 150 so the good docking score, the range between 0 to 10 kilocalorie per mole. High negative score denotes the strong bonding, while a less negative or positive score denotes a weak binding. The best 3 compounds A3, B5, and C5, which having the best docking score, their process to 3D picture are depicted in figures. First figure is show the docking pattern of compound A3. And second one, the rocking picture of the compound B5, and third one, the rocking pattern of compound C5. This is the references. Thank you, ma'am.
Thank you so much, sir. The forum is now open. I would request our guests to kindly interrogate for the queries.
So, Dr. Vikash, am I audible to you?
Yes, sir.
So, can you tell me like what is the final outcome from this project?
Final outcome, we study for 1,5-benzothiazepine compound selected for molecular docking study for anticonvulsant activity. By the literature survey, we found that the third position and fifth position of benzothiazepine nuclear. So the compounds are best compound for anticonvulsant activity. So we try the by molecular tracking perform and find out the best fit compound which show the more activity.
So all this is like computational data, right? Now you are planning to go for the synthesis followed by the biological evolution in probably in vitro and in vivo models, right?
Yes, sir.
Okay, okay, Dr. Vikas. Yeah, I don't have any further questions. Thank you.
Okay, thank you, sir.
Thank you so much, sir. I would request our next participant, Deepak Mishra. Deepak Mishra, to please join. Okay. Is Jyoti Singh available? Can you hear me, Jyoti? Do we have Mansi Jain with us? Do we have Runali? Jyoti Singh, can you hear me? Please unmute yourself.
Jyoti Singh, please accept panelist request.
Good afternoon, sir. I will give the presentation after some minutes. I need some time.
Sorry, Jyoti, you're not audible. Can you repeat?
Good afternoon, ma'am. I will give the presentation after 20 minutes. I need some time. Am I audible to you?
Yes, Jyoti.
There is some issue in the system. I need some time. So I will join after 20 minutes. Means I will give the PPT after 20 minutes.
Okay, Jyoti. Next we have Shrishti Shandalia with us. Can you hear me? Shrishti, can you hear me? Kindly unmute yourself.
Srishti, please unmute. You can unmute. Srishti, can you hear us?
I think so. She's facing some technical issues.
Srishti, can you please raise your hand? Yes, yes. Raise. Please raise your hand. Srishti, now please mute.
Good afternoon, ma'am.
Are you able to join?
Yes, ma'am, now I am able to join.
Jyoti, kindly turn the camera and share your presentation, please.
Yes, ma'am. Ma'am, is the presentation visible to you?
Not now. Yes, your video is now visible. Can you please share your screen and presentation?
Ma'am.
Your screen is sharing. Now you can start.
Yes, ma'am. Good afternoon, everyone. My name is Jyoti Singh. I am from Pharmaceutical Science and Technology Department, B Farm second year, and I'm from Triple M University of Technology in Gorakhpur. So my topic for presentation is brown adipose tissue and glucose starvation affecting tumor growth. So, firstly, I would like to discuss about cancer. That is a metabolic alteration which that are hallmark of various cancers and tumors. And tumor cells needs a lot of glucose to proliferate number of cells and division as they so very fast division. So they needs a lot of glucose molecules. So for this, they used to under aerobic condition, tumor cells use the glucose lactate pathway to generate ATP, and this pathway is also known as Warburg effect, and given by Otto Warburg. And now I would like to discuss about BAT. Actually, we have to control tumor and various cancers so that its proliferation we have to stop this. So I would like to discuss about BAT, that is brown adipose tissue, that play a key role in the thermogenesis process and energy expenditure in the body, so that the cancer cells would not get sufficient amount of glucose, and in insufficient amount of glucose they would not be able to proliferate and not be able to divide. So brown adipose tissue is a adipose tissue which is found in the suprarenal, supraadrenal and perirenal, supraclavicular region of the neck and the back side of the body. And but it is found in activated form. We need to activate it, and basically it is found in mammalian body, and we need 18F fluorodeoxyglucose position emission tomography and computed tomography for the scanning of and the distribution of bat. So the bat needs to be get activated. It is activated by cold acclimatization and drugs. So and also died. So activated bat is able to generate heat. Non-activated bat don't produces heat. So bat shows non-shivering thermogenesis mediated by UCP1. UCP1 is protein that is found in the inner mitochondrial membrane of the brown adipose tissue, and actually norepinephrine that is released from the sympathetic nervous system is able to activate beta adrenergic receptor that is present on the brown adipose tissues membrane. So this UCP protein that is present on the inner mitochondrial membrane is able to activate and do the process of thermogenesis, and muscles are not involved in this. So that's why this process is also known as non-shivering thermogenesis, because no muscle contraction is involved in this, and sequential activation of enzymes occur, that is adenylate cyclase, protein kinase, and hormone lipase, and thus fatty acids are released which are able to activate the UCP protein present on the inner mitochondrial membrane. And when the UCP protein get activated, it dissipates the proton gradient without generating ATP, resulting in the expenditure of energy as heat. So this kind of activation with cold activated bat is essential in the tumor suppression. As we can see in the figure, that is cold when in presence of less temperature or cold exposure, sympathetic nervous system is get activated, and the adrenergic receptor present on the inner mitochondrial membrane of the brown adipose
Jyoti,
tissue
Jyoti,
get
sorry to
activated.
interrupt you. Jyoti, can you hear me?
Yes, sir, I can hear you.
I'm sorry to interrupt you, but your slides are not moving actually. I don't know whether it is in my screen or in organizer team screen as well, but I don't see any changes in your slides. It is only the, I think, the
Sir,
first
for me
slide.
also, her slides are not changing, sir.
So that's why, how we can evaluate then if we don't know what is the content. We are just listening what you're telling, but we are not able to see what is there in the slides.
My slide are not visible.
No, they are first slide, only first slide is
Only
coming.
first slide.
The slide is not, you have to, enable editing, I think, and you have to come in the full mode. You have not selected the enable editing. Just see on the top panel, enable editing, and then you have to come into the full screen. Then only you can change the slides.
Okay, sir.
Now your
Unfortunately,
slides are changing. Other,
So now it is changing.
yes. I think you
okay.
forgot to change the slides in between.
Is it now visible?
Now it is moving. So please proceed.
Sir, from beginning or from the slide on which I'm speaking?
Just try to scroll slowly from the first slide so that sir can have a little view about it.
Okay, sir. Is it visible to you? The first slide is about
Yes,
introduction.
it is fine, Jyoti. Please go ahead.
Okay, sir. I repeat again the whole slides.
Yeah, just quickly, don't take much time. Just quickly you proceed.
Okay, sir. So this presentation is about brown adipose tissue and glucose starvation affecting tumor growth. So basically, we are talking about cancer cells and its suppression, that is tumor cells. So we know that cancer cells proliferate very fast, and we need to stop their proliferation and metastasis. So that's why we are talking about BAD, that is brown adipose tissue, which is an essential factor that helps in reduction of glucose, so that tumor cells don't proliferate. We know that tumor cells, so under aerobic conditions, they show glucose fermentation lactate pathway to generate ATP, and they show very fast division. So this effect is also known as Warburg effect, given by Otto Warburg. And so So I would like to discuss about BAT. That is it visible to you?
Yes, please go ahead. I will interrupt you if there is any issue. Please proceed.
Yes, sir. So brown adipose tissue is a tissue that is that play a very important role in non-shivering thermogenesis. That is, without muscle contraction, heat production takes place in the body and energy expenditure occurs. So this tissue is very important, and it is scattered in the supraclavicle, paravertebral, pericardial, and suprarenal regions of rodents also, and in mammals, mainly rodents, and in small babies we can say, and in humans by growing age it regresses, but it is found in the neck region, and it needs to be activated because it is mainly activated by cold. So the presence of brown adipose tissue had detected by 18F fluorodeoxyglucose PET-CT scan, position emission tomography, that is most frequently used method to detect the activity and presence of distribution of BAT in human body. Since activated BAT is good at intaking glucose, so that's why it can be useful in detecting various kind of cancers. So now I need to tell about that is how can be BAT activated. So BAT is activated by many ways, but the most common ways are cold acclimatization, that is reducing the temperature, and by diet, and by drugs and therapeutics. So activated BAT, when BAT gets activated, so it's so non-shivering thermogenesis. Without muscle contraction, heat is dissipated, and this is done by UCP1 protein that is present on the inner mitochondrial membrane of brown adipose tissue, and this UCP protein is regulated through sympathetic nervous system. So actually, sympathetic nervous system stimulates norepinephrine hormone, and that norepinephrine hormone activates beta adrenergic receptor present on the brown adipose tissue's membrane. So this UCP protein is responsible for the thermogenesis caused by the brown adipose tissue. As we can see in the figure, that is cold activated, that sympathetic nervous system, and the norepinephrine released, and fat browning of fat also occur, which converts into brown adipose tissue, and again the thermogenesis process goes on. So UCP1 increases. UCP1 is a protein that is mostly found in the brown adipose tissue. Unlike normal cells or VAT, it is not present most amount in VAT, but in BAT it is present in maximum amount, and this UCP1 protein is responsible for the thermoregulation and heat emergence. So that's why under cold it is very effective to generate this. So now effect of cold, what is the effect of cold activated BAT on glucose? Means why we need to activate BAT, and what is the relation between glucose and BAT, activated cold BAT? So from scientists and researchers have done experiments, and from the 18 FDG PT CT scan, it was clear that cold activated BAT has the ability to take significant amount of glucose. So from tumor cells, if glucose will not be get to them, then the proliferation would stop. So that's why this experiment was successful, and it's tell about that under thermoneutral conditions, 18 FDG distribution was mainly occurring tumor tissues. That is, glucose was getting by tumor cells, and they were proliferating under thermoneutral conditions. But when activated BAT, that BAT was activated under cold.
Jyoti, your time is exceeding, so I will ask the judge to please interrogate her with the questions.
So yeah, Jyoti, can you hear me? Your voice is not audible. Can you please unmute yourself? You are muted right now. Please unmute. Can you please unmute yourself, Jyoti?
Yes, sir.
Yeah. Okay, so what I can see from your work, you have tried to investigate the role of brown adipose tissue in the tumor suppression, right? And
Yes.
you are proposing the use of cold induce activation of BAT, right?
Yes, sir.
So do you have any data on this, or you are just proposing a theoretical concept here?
No, sir. I have data done by scientists and researchers, and I have
Okay.
references also. In the last of the slide, I have added references.
Okay. So this is not your work, right? You are an undergrad student or a master student?
Sir, I am undergraduate student.
Oh, you are an undergrad student.
Second
You are
year,
just
BPharm
proposing a
second year.
theoretical work. Okay. So what is the mechanism of cold induced BAT activation?
So actually, cold-induced BAT activation activates BAT through that non-shivering thermogenesis occurs by the UCP protein, that is uncoupling protein, present on the inner mitochondrial membrane of the brown adipose tissue. And when it's get activated, then various kind of enzymes get activated, and the fatty acids lipase enzyme get activated, which activates the UCP1 to non-shivering thermogenesis. And in that case, when BAT get activated,
So
then
do you
it
know how much
uptakes.
percent of different tissues, how much percent of brown adipose tissue in comparison to like other adipose tissue?
Sir, in total body, 0.5% by weight of total body percent, BAT is present.
Okay, so do you know what are the pathways of apoptosis, because you are proposing something on cancer? So do you have any idea about the pathways, or
Sir,
are you aware about the pathways of cancer? What are the pathways of cancer, extrinsic, intrinsic pathway? Do you have any idea? What are caspases?
apoptosis is a programmed cell death. I
Mhm.
don't know much
What are
about it.
caspases? Do you know about the caspases?
Yes, sir, it is used for activating BAT. I am
No, that is not correct. So do you know the difference between caspase 8 and caspase 9?
sorry, sir.
Okay, problem. No problem, Jyoti. So I think being an undergrad, this is a nice attempt, I would say. So thank you so much. Keep on continuing the good work. Okay. Thank you.
Okay, sir. Okay, thank you, sir.
All the best.
Thank you, sir.
Jyoti, please unshare your screen, and Deepak, please be ready.
Okay, sir.
Kindly.
Ma'am, shall I join?
Deepak, kindly share your screen, please.
Ma'am, am I visible?
You are visible. Your screen is not visible yet now.
Okay, ma'am. I'm sharing my screen, ma'am. Ma'am, is my screen shared?
Yes, your screen is visible, Deepak. You may start now.
Thank you everyone for providing me this golden opportunity. Here I am to present my topic, or a role of autophagy in cancer, and overview.
Hello, can you hear me?
Yes, sir.
So your name is not there in the title slide. Can you please go back? May I know
So
your-
we were told to not put the name.
Okay, no issues. What's your name?
So my name is Deepak Mishra, and currently I'm assistant professor.
Okay, Deepak.
Yes, sir.
Okay, Deepak, please proceed.
This is my table of content. Uh, so the topic is autophagy and its role in cancer. So basically, the autophagy is nothing but the fasting that we do. It is also a type of autophagy only. So auto means self, and phagy means phagocytosis. That is the eating of cells. So here the cells are being degraded by the lysosome. So we are learning from our childhood that is lysosome are the suicidal bag of the cells, so that are used for the process of autophagy. And yes, this autophagy is very much responsible for the progression of cancer as well as the inhibition of cancer. So that we will be discussing in this slides. And mainly there are three types of autophagic pathways, that is microautophagy, macroautophagy, and chaperone mediated autophagy. And this is the basic mechanism that takes place during the process of autophagy. And the first one is the autophagy initiation. that is rolled down with the membrane nucleation and phagophore formation. The next one that is phagophore expansion, and the last one that is fusion with lysosome. That is the fusion with lysosome results in the formation of autolysosome, and at last the degradation takes place with the help of lysosome. These are the types of autophagy. I have compiled the two by comparing them. That is microautophagy versus macroautophagy. Here in the macroautophagy, the autophagosome, phagosome is directly formed with the help of phagophore, and the extension of the phagophore helps in the formation of autophagosome. Whereas in microautophagy, it is the internal process where the autophagic body is formed inside the vacuoles, and this is helped by the permease enzymes. And the third one, that is the last one, is the chaperone-mediated autophagy. Here also the process is same, but there is the role of chaperone. That is the HSC heat shock current 70 that helps in providing the substrate to the autophagic process, and at last the autophagic degradation takes place by the help of CMA. That is chaperone-mediated autophagy. And the next one is the most important one of them all, that is the regulation of autophagy. So these are the factors or the proteins that are responsible for the regulation of autophagy. The first one of them is autophagy related proteins, that is also known as ATG. Next one goes with the Bacillin 1, which is the central regulator. This helps in the proper regulation of the autophagy. The next one is the ULK ATG1 kinase core complex. This helps in the formation of complex, that is autophagosome formation. And the next one is the LC3 ATG8 conjugation system. This is also one of the characteristic property of the mammalian shell that helps in the autophagosome formation. And the next one is the role of autophagy in cancer treatment. So here, as I have already discussed, there is the dual role of autophagy in the cancer therapy. That is, it can also mediate the cancer progression or promotion, and also it has the capability in the suppression of tumorigenesis. So here the mediation takes place by the promotion of survival and growth of established tumor by removing the toxic reactive oxygen species, that is ROS, which is responsible for the degradation of cells. But here the ROS is neglected, and that's why the progression of tumor cells takes place. The next one, the suppression is helped by the elimination of damaged proteins and organelles by the help of autophagy, which is the major role of the autophagy. That is the self-degradation of the damaged cells or unwanted cells. Yes, and then I have included this table that shows the gene and the role in the autophagy. So these are the different genes, and their roles has been mentioned in the table.
Your time is up. Please kindly unmute yourself again and please defend the questions.
Deepak, can you listen me?
Yes, I can hear you.
So yeah, what I see, like most of this work is theoretical only, right? Yeah, have
Yes,
you
sir.
performed any experiment or not?
Not on this topic, but I have my master's topic, but it has not been published. That's why I did not mention that.
Yeah, but it doesn't matter, if the work is not published, you could have presented, because research data is always, you are already a faculty, right? This is what I can see from your profile, right?
Yes, I have recently joined. I am 2023 pass out, sir.
So presenting a research finding is always, have more weightage than
Sure,
presenting
sir.
something which is theoretical in nature, right?
No, that's
Yeah, if you're an undergrad student, it is understood, in undergrad, we don't have access to, like project and, like
yes.
lab or, like animal house facility. So all these things are issues with the undergrad student, but being a master's student, you already have data. You generally
Sir,
expect that you should present. Yeah.
you are not audible to us.
Sir is audible to me.
Okay. So I think you have some issues, because Deepak can hear me actually. Can you hear me?
Yes, sir. You are audible to me.
Okay. Okay, good. But your video is not coming, Deepak. Can you stop sharing your screen?
Now?
Yeah, now it is better. Now I can see you. So
Okay, sir.
I can see that you have a good theoretical knowledge, Deepak. But try to, like present the research data whenever you are going to some meetings or
For
seminars.
sure.
Okay, and we always want that people should present unpublished data. Later on you can publish it. There is no problem.
Okay.
Okay, yeah. So
We'll take that in mind, sir.
okay, okay, but so do you have any idea like about the this significance of autophagy, like how you can activate the autophagy?
So
Are
the
there any
activation
ways?
of, yes, sir, there are the different proteins that such as ULK1 protein. It is responsible for the initiation of the
Can you tell me
initiation of the.
something? Can you tell me something in our culture, in our Indian culture,
Yes, sir, it
which can
is.
help into to activate the autophagy?
Yes, sir, the
How can
fasting. The we have
fast?
a we have a
Okay.
5,000 years of tradition that we keep fasting, and in the modern world it is known as intermittent fasting,
So
but
do you
it
know
has
about?
been followed.
Okay. So do you know what is the mechanism of like autophagy? Like what does autophagy does
Yes,
actually?
I know.
To the-
So basically, as we keep fasting, the energy level gets decreased, and the energy is uptaken by the well developed cells, and the less developed or damaged cells gets deprived of the energy, and that leads to the damage of the cancerous cell, or we can say the damaged cells. That helps in the proper maintenance of our homeostasis.
Okay, I got you, got you. Thank you, Deepak. So continue the good work and wish you all the
For
best
sure,
from
sir.
my side.
Thank you so much, sir. My pleasure, sir. Thank you.
I would request Divyanshi to please join and share her screen.
Yes, ma'am. I'll sharing the screen.
Please on your video, Divyanshi.
Ma'am, it's visible.
Yes, it's visible.
Ma'am, screen.
Screen is not visible yet now. It's visible.
Okay.
Yes, Divyanshi.
A very good afternoon to all present here. My topic is fighting polycystic ovarian syndrome with the herbs and phytochemical. So, polycystic ovarian syndrome is one of the most important gynecological disorder nowadays. According to WHO, nowadays in worldwide, around 116 million women are affected. This is the main reason of in women due to which they leads to infertility. Other symptoms of polycystic ovarian syndrome includes menstrual disorder, hirsutism, acne, bladness, and infertility. There are also changes in endocrine hormones which leads to decreased androgen level, decreased progesterone level, and other metabolic disorders like insulin resistance, diabetes, dyslipidemia, and type 2 diabetes. The basic reason behind the PCOS is the disorder of gonadotropin secreting hormone from the pituitary gland, which leads to the increased secretion of androgen hormone. Polycystic ovarian syndrome is an oligogenic disorder caused by the various genetic and environmental factors that I have showed in figure number 1. Is this key? Basically, it is due to various lifestyle disorders, sedentary lifestyle, or maybe oxidative stress on some environmental factors. There are some evidence that women with PCOS can benefit from the supplementary therapies such as herbal remedies and nutritional supplements. Nutrition supplements have demonstrated benefit in PCOS suffers and risk reduction for the similar issues in other population. Homocysteine and oxidative stress are also helpful in are also noticed in elevated PCOS patients. The use of herbal remedies the use of herbal remedies in PCOS helps in promoting the decreasing the androgen level, which was increased. Different herbs are there which helps in treating PCOS like curcuma, longa, licorice, chasteberry, Chinese chastity tree, etc. Currently, in pharmacological management, there are clomiphene citrate, metformin, and tamoxifen are widely used. However, all these pharmacological management with the long-term uses having many complications like gastrointestinal disturbance, weight gain, insulin resistance increase, less compliance, and poor efficacy. With regards to these side effects, I propose a work I propose plant-based drug, especially containing estrogen, would be helpful for the PCOS. This is the basic pathophysiology of PCOS, respecting anti-androgen component as a promising treatment strategy. The basic mechanism of pathophysiology of PCOS is brain release gonadotropin releasing hormone, that is GnRH, which further help in the secretion of more androgen and male sex hormone, that is testosterone. Due to increased testosterone in the ovary, there is increased luteinizing hormone and decreased follicular stimulating hormone, which result in the polycystic ovarian syndrome, hirsutism, that is male-like hair patterns, acne, and insulin resistance. So with the help of some herbal medications, yeah, herbs, we can decrease this increased hormone, that is testosterone, or we can increase follicular stimulating hormone, which help in treating these symptoms. And along with this pathway, there is one another pathway that due to increased follicular stimulating hormone and luteinizing hormone, there is an increase in testosterone level due to disturbed signaling pathways. We use a herb which can promote or which can manage this insulin pathway, that would also be helpful in treating the PCOS. These are the available treatments that are available. The pharmacological treatment are androgen receptor antagonist, oral contraceptives, insulin sensitizer. In non-pharmacological management, currently there is some physical exercise and green teas are available, and some dietary supplements. Herbs for the treatment of PCOS. I have collected a data for some herbs like curcumin. Curcumin is basically found in turmeric. It is the herb which helps in manage the elevated hormone levels and manage elevated insulin level in PCOS. Curcumin, in clinical trial, it was found that the 1300 mg of curcumin in 3 times helps in women who are suffering from PCOS. Basic mechanism behind the act of curcumin is curcumin reduce the inflammatory cytokines which are responsible for the PCOS. It functions by preventing the hepatic glucose, lowering the intestinal glucose absorption, and enhancing the glucose metabolism. It also works by preventing the expression of vascular endothelium growth factor, a progenic factor that is
Divyanshi, your time is up. Sir, please proceed with the questions.
Divyanshi, can you please unmute yourself? Divyanshi, can you please unmute yourself? You are muted right now. No, please unmute. Kindly unmute yourself.
Divyanshi, please try to unmute yourself.
Ma'am, I've already unmute myself.
Now
Okay.
I can listen to you. Okay. So you have mentioned, about the PCOS, right?
Sir, your voice is not audible.
Hello, can you hear me?
Sir, it's coming very low.
Can you hear me now, Divyanshi?
Yes, sir.
Okay, so you have presented the work related to PCOS, right?
Yes, sir.
And you are talking about few of the herbs, which are there in use. for the
Which I have.
PCOS.
Yes, sir.
So you have also spoke about the curcumin, right?
Yes, sir.
Can you tell me what is the mechanism of curcumin by which it is helpful in the treatment of PCOS?
Yes, sir. So actually, basically, that from the literature we have collected, the curcumin basic is key, that is reducing the inflammatory cytokines, which are ultimately raising the hormonal levels like androgens and progesterone. So curcumin will act by decreasing.
How curcumin is modulating the hormonal levels? That I don't understand. I know that curcumin is a very good molecule. It has,
Yes.
a lot of,
So it
anti-inflammatory
decrease the
activities.
increased progesterone level.
How?
So
I don't know about that because there are drugs in the market, which can, modulate the hormonal levels, right?
yes,
But
sir.
can curcumin also modulate the hormone levels?
Yes,
And
sir.
how? What is the mechanism?
Sir, it basically act by decreasing the secretion of progesterone through pituitary gland, and also reduce the oxidative stress in the body.
No, look, there are a lot of antioxidants. So do you mean to say that all the antioxidants, which are there out, they can, modulate the hormonal level by decreasing the oxidative stress? Is that what you mean to say?
Yes, sir.
No. That is not correct. That means all the antioxidant will modulate the hormonal levels? No. Hormonal drugs are different category of drugs, they
Yes,
work by
sir.
different mechanisms. For that you need to understand the whole physiology, how the hormone secretion is regulated and controlled. Okay.
Yes, sir.
So which is the master gland? Do you know that?
Yes, sir. Pituitary gland.
Okay. And then how the levels of estrogen and progesterone they are regulated?
So through pituitary gland.
How? What is the pathway? Okay, no issues, no issues. So that's all, Divyanshi, from my side, and
Yes.
wish you all the best.
Oh, thank you, sir.
Mansi Jain, are you ready?
Yes, ma'am, I'm ready.
Please share your screen and turn the video on.
It is visible, ma'am. It is visible, ma'am. Ma'am,
Yes, yes, your screen is also visible. You can start now.
okay. Good afternoon, everyone. My name is Mansi Jain. I'm a junior research fellow. Good afternoon, everyone. I am Mansi Jain. I am junior research fellow under the supervision of Dr. Neeraj Mishra sir and Dr. Vikas Pandey sir in the Department of Pharmaceutics, MIT Institute of Pharmacy, MIT University Gwalior. So my topic is animal preclinical models for inflammatory bowel disease.
Mansi, kindly unmute yourself. You're on mute.
Mom, there is some problem with my slides. They are not moving.
Please do not keep it in a presentation mode.
Ma'am, please give me some time.
Okay, Mansi. Yes, Mansi, share it in this form also, not an issue. Please start.
Give me some
Yeah.
time. Now. My topic is animal preclinical model for inflammatory bowel disease. Now it's not moving. मैंने कुछ नहीं दबाया है.
Mansi, do not keep it in a presentation mode. Please continue.
Okay, sorry to disturb you, ma'am. So let's start with the introduction. Inflammatory bowel disease is a group of chronic inflammatory conditions that affect the digestive tract. There are two types of bowel disease: Crohn's disease and ulcerative colitis. I'm choose this topic because I'm already working on this topic. About 10 million people are worldwide affected with this disease. especially youngsters are suffer from this disease because of their unhygienic lifestyle and overtaking of fast food. Crohn's disease, difference between the disease and ulcerative colitis is that Crohn's disease affect the all GIT portions, but in other ulcerative colitis, it's only affect the large intestine. It's not moving.
इसे मूव करें
Mhm.
ये सी देखो ये सी ऊपर वाला ऊपर सबसे पहला पहला बटन जो ऊपर है पोस्ट
It's not moving.
Mansi, please do not sharing the
Ma'am,
presentation.
ma'am,
Not like
there is internet problem.
हाँ, it's okay. It's okay.
Okay.
यहीं से start
So-
from here itself. Yeah, please.
Okay. Challenges with the animal model. Ethical concern, limited human relevance, variability in result, high cost, limited variability of animal, and importance of animal-free IBD models. So, ethical consideration, human relevance, precision and reproducibility, and cost effective. Now discuss about the animal-free model used in IBD. So, in vitro cell culture model, microfluidic system, co-culture system, biopsy-based ex vivo model, human on a chip model, bioprinting model. In the diagram, there is a difference between traditional research and modern research for preclinical study of drugs. In vitro cell culture model. In vitro cell culture model or cell culture model involving growing human intestinal cell, often from the biopsy sample of IBD patient in the lab. Researchers can use these cell culture cell line like CACO2 study for the inflammatory bowel disease. Microfluidic system. Microfluidic system, also known as a lab-on-chip system, are involving tool in the biochemical research that enable to manipulation and control of the small volume of fluid within micro-scale channel. These systems often a wide range of application, including the study of, of disease, inflammatory bowel disease, human on chip model. These model combine multiple organ on chip system to replicate the interaction between the different organ in the human body. These models aim to recreate the physiological and pathological condition of human organ and tissue on micro scale device. While human on a chip model have been developed for various applications, including drug testing and disease modeling, they hold promise in the study of inflammatory bowel disease. Biopsy ex vivo model. Biopsy ex vivo model provide a bridge between the in vitro and in vivo research. These models involve the extraction of tissue sample typically obtained through a biopsy procedure and then subjecting these sample to various experimental conditions to better understand the tissue characteristics, response to treatment, and underlying mechanism of disease. Bioprinting system or bioprinting model. Bioprinting in this model, it is also known as a 3D model. 3D bioprinting technique are create tissue models that mimic the structure and function of the intestine, enable the study of IBD in a controlled. Am I audible, ma'am? Am I audible?
Yes, yes, you're audible.
Yeah. So
Dear, please conclude. Your time is
co-
exceeding.
co- okay. So conclusion, we discuss alternative experimental models, and for the pharmacokinetics could be evaluated using the in vitro models that stimulate the digestive system because of their low cost and simple anatomy. Vertebrate model are widely used in the genetic research and toxicity testing. Therefore, these non-animal experiment methods can help with the moral goal of reducing the use of animal in the scientific research. Thank you, sir.
Thank you, Mansi, for finishing in time. So, do you know about the pathophysiology of the IBG and the prevalence of this
Yes,
disorder?
sir. Yes, sir.
Just be brief, okay? Don't discuss in detail. Just in one line, or two
What?
lines.
Yes, sir. It mainly it is a yeah. There is two types of IBD. So first one is Crohn's disease that cause the inflammation like swelling, pain, abdominal pain, and specific diarrhea in the whole GIT. But
Mansi,
in
you
the
are not getting my question, you know. I have
yeah,
asked you the pathophysiology, okay? What is the pathophysiology of IBD? Okay, just
sir,
forget
it's
about
a
it, okay?
etiology. Sir, it's a etiology is not fine.
Okay, okay, Mansi. So, okay, just one suggestion, you know, for you. In any of the slides, you know, you have not mentioned the references. If you go back to your slides, you are presenting different techniques, you know, like alternative
Yeah.
techniques, you know, which can be helpful, you know, in the,
Yes.
like, in screening the, identifying the new targets, or, developing new, ways to study the IBD. But if you, in none of your slides, the references are completely missing. So all this content you might have taken from somewhere, right? From literature you might have referred.
Yes, sir.
If
Yes,
you're
sir.
not quoting the references, that means you are doing injustice to the people, who have done this work. So it is
Yes,
always
sir.
recommended. You're a master student or undergrad?
Sir, I'm master student.
So you should know that, right? At
Yeah.
undergrad level, we don't expect this from the student, but at master student, you should understand the importance of literature review and how to quote the references. So that's
Yes,
a
sir.
suggestion for you for future, okay?
Yeah. Thank you very much, sir. I'm so
Thank you.
sorry.
Wish you all the best.
Thank you very much, sir.
Ghazala, please join quickly. Mansi, please unshare your screen. Ghazala, please start your video. Ghazala, please speak.
Ma'am, my video is visible. Hello.
Your screen is visible. Can you speak up, please? Please turn your camera also.
Now I'm visible, ma'am.
Thank you so much, Ghazala.
Ma'am, I am visible now.
Kindly speak little loudly. You are not audible.
Am I audible now, ma'am?
Yes, please. Please start.
Okay.
Very good afternoon, respected judge and dear participants. The title of my presentation is A Contemporary Review on In Silico Approaches for Drug Discovery, an Alternative to Animal Experiments, and the code is AIP/AUUP AEE/3/11.
Any issues, Gazala?
Yes, ma'am. Actually, the slide is not moving. Okay. Is it visible, ma'am?
Your slide is not moving.
Right now, ma'am?
Try to share it again.
Okay.
Stop the sharing and try to reshare it again. And please
Sure,
do
ma'am.
not do the presentation mode.
Is it visible, ma'am?
Yeah, it's visible. It's
Ma'am, is it
moving
visible?
also. Yes, yes.
Okay.
Please continue.
Okay, ma'am. Okay. Okay, the introduction is that in silico study is also known as computer-aided drug design. It is a method that played a major role in the drug development as well as in the discovery of the drug. By using these computational techniques in the drug discovery as well as the development process is rapidly gaining the popularity as well as the implementations and appreciations for the development of drug discovery. Different terms has been described to apply these techniques like computer-aided drug design, computer-aided molecular docking, computer-aided molecular design, and in silico studies. The CAD entails use of computing powers so that the drug discovery and development process can be achieved, and it's leverage the chemicals and biological informations about the ligands as well as the targets to identify and optimize new drugs. It also designed with the help of in silico filters that eliminate the compounds with undesirable properties, like those drugs which have the poor bioavailability and they are not active. It also improved the absorption, distribution, metabolism, excretion, as well as it also helps in the removing of toxicity of drugs. And it select the most promising candidates by using the computer-aided drug design or in silico study. Next is classification. Computer-aided drug design is divided into two parts. That is basically the structure-based drug design and ligand-based drug design. Further, the structure-based drug design includes molecular docking, inverse docking, and virtual screening like structure-based virtual screening, pharmacophore, or ligand-based virtual screening. In ligand-based drug design, the computer-aided design includes the chemical similarity as well as the quantitative structure activity relationship. The structure-based computer-aided drug design relies on the ability that determines and analyze 3D structures of biological molecules. Means the core hypothesis of this approach is that the molecule's ability to interact with a specific protein and exert a desired biological effect that depends on the ability to favorably interact with a particular binding site as well as on the proteins. Like in structure-based drug design, the virtual screening is used that discover the new drug candidates from various different chemicals scaffolds by searching the commercial, public, or private three-dimensional chemical structures databases. So it is intended to reduce the size of the chemical space and thereby allow the focus on more promising the candidates for the lab discovery and optimization. Like the overall is that the central goal of the structure-based in silico studies is to design the compounds that bind directly to the target, that is with the large reduction in the free energy. So if the compound will tightly bind to the target, then it will achieve the more therapeutic effect, as well as it also improve the ADME as well as the toxicity if the drug will bind on the target sites. So, like Swiss ADME, that is a web tool that gives the free access to a pool, a fast and yet robust predictive model for the physicochemical properties as well as the pharmacokinetic and drug likeness and medicinal chemistry. Another software that is automated docking tool that is also designed to predict how the small molecules and drug candidates bind to our receptors. So if the particular molecule will bind to the target site specifically so that we can improve the toxicity studies, the toxicity of that particular molecule, if it bind to the target site specifically, and that will provide the desired pharmacological action. The next is ligand-based drug design. Ligand-based drug design exploits the knowledge of known active as well as the inactive molecules through the chemical similarity searches on construction of productive quantitative structure activity relation to the models, and the ligand-based drug design is generally preferred when no or little structures information is available. So, like for example, often they are used for the membrane protein targets. And in the ligand-based drug design, the
Ghazala, your time is up. Please unmute yourself and be ready for the questions.
Hello. Ghazala, can you hear me? Hello. Am I audible?
hello.
Yes, yes. Ghazala, can you hear me?
Yes, sir.
So, okay. So, yeah. So you're presenting, but you have to be quick when you are having a definite timeline, so you need to, make sure that you are meeting the, desired timeline which has been assigned to you. Okay, that's fine. Again, the same problem which I observed, in case of previous presenter, none of your slides are quoting the references. So that means, whatever you are writing over here, is your like own language, or, is your own work, I would say, rather, instead of, like taken from somewhere. So
It
not
is taken
quoting
from
the
somewhere.
literature is not good. Okay. You should give due credit to the authors to which this particular work belongs to. Okay. That is one thing.
Okay, sir.
Second thing, do you know the difference between G score and D score? We're talking about the docking studies and all those things, right? Computational tools.
Yes, sir.
So any idea about the difference between glide score or the docking score?
No, sir.
How they are different from each other?
No, sir.
Okay. You're an undergrad or a master student?
So I'm a PhD scholar.
You're a PhD scholar. Are you using the computational tools in your PhD?
Sorry, sir.
Are you using computational tools in your PhD?
No, sir.
Okay. Okay, fine, Ghazala. Wish you all the best. Thank you.
Okay, sir. Thank you.
Ghazala, please quickly unshare your screen, and I'll request Rabia to unmute herself and start share the screen, and please turn your video on. Rabia, can you hear me? Please try to unmute yourself.
Yes, ma'am. I'll try.
please turn your video on.
Ma'am, I'm audible to you all?
Rabia, please turn your video on.
Okay, ma'am, we'll try. Yes.
Yes, you are visible right now. Please try to share your screen.
Okay, ma'am, we'll try. Okay. Ma'am, oh, sorry. Good afternoon, all of you. Today I'm going to present my topic is zebrafish as a multifaceted model for
Rabia, your screen
disease.
is not sharing. Please share it.
Okay, ma'am. Screen share.
It's visible right now. Please start.
Okay. Okay. Good afternoon, everyone. My topic is zebrafish as a multifaceted model for disease research and nanomedicine insight, innovation, and future horizon. As we all know that there is a various animal in research fields like guinea pig, rats, monkey, etc. But now, apart from this, we have good vertebrate model like zebrafish. So let's start the zebrafish model introduction. So zebrafish is a Danio rareo. Zebrafish is another name is Danio rareo, is a popular model organism in scientific research due to its numerous advantages. So over the past 10 years, we have become the increasingly popular tool for scientific conducting biomedical studies and other researches. Due to their its zebrafish characteristic like transparent embryos allowing for easy observation for developmental process. So researchers may can use zebrafish to investigate a wide range of topics for development biology to neuroscience, making it a versatile and powerful model in scientific research. So this is the life cycle of zebrafish. We can see clearly here why it's called zebrafish. Zebrafish due to call for appearance like zebra. So and life cycle of zebrafish. So there is a zebrafish developed rapidly from one
Rabia,
cell
your
like
screen is not
yes,
moving.
ma'am. Slide is not moving.
Yes,
Okay,
please try to
ma'am. Is
move.
this
No, it's not moving.
ma'am? Wait a minute. Sorry. Ma'am, my slide is visible right now.
It's visible.
Okay. Now slide. This slide is visible right now, ma'am.
Yes, yes, please continue.
Okay. This is the life cycle of zebrafish. Zebrafish rapidly from one cell zygote that sit on the top of large yolk cell. And these are three steps. And after three steps, zebrafish form gastrulation, What is the mean of gastrulation here? Gastrulation beings a critically embryo development for the before blastoma. So here after blastula makes form oogenesis, and now for begin approximately 6 hour post fertilization hatching at 2 days as a free swimming larva, and then zebrafish reach sexually mature around 3 months of age and can live for up to 5 years. So here the zebrafish model developing various diseases like cancer, cardiovascular disease, and neurological disorders. Cancer, zebrafish are used to study cancer development, tumor potential, and drug treatments. There are numerous drugs that have been evaluated zebrafish cancer, like doxorubicin and other anti-cancerous drugs. And now cardiovascular disease have also developed in this model, through this model, like heart regeneration, heart development, and various heart-related condition. Example, proparanol.
Bhavya, your slide is again not moving.
Ma'am, there is some issues, I think. I'm trying. Ma'am, now slide is visible right now?
Yeah.
Hello. Okay, ma'am. Sorry, there is some network issue. Okay. So, zebrafish model developing neurological disorders, and there are some few examples on the drugs like 1-phenyl 2-thiourea, clozapine, aminopyridine, and 1-methyl 1-phenyl tetradonisol, and pentylene tetrazoles. So this is the genetically engineered fish lines, and this diagram showing how cancer develop in a zebrafish model. A indicating the after breeding and mating the zebrafish, and then founding the transgenes, stable, individual, or expression of gene interest. And after this, if some genes are found disables, then disqualified by the knockout, knockdown, or knock-in. So there is a two process like wild type and transgenic or mutant embryos, and find C, and this is the stage where the transplantation of tumor in zebrafish larva for the drug screening and functional analysis, and then find juvenile fish and tumor progressions in zebrafish. So here the another model development disorders in zebrafish models, like some.
Slides not changing.
Bhavya, your time is up. Please unmute for the questions.
Okay, ma'am.
Can you hear me?
Yes, sir. Yes, sir.
Unfortunately, your slide was.
Sorry, sir. I am not hearing. Please.
Can you hear me now, Rabia?
Yes, sir. Yes, sir.
So unfortunately, your slides were not moving. So I was not able to.
Sorry, sir. There is some network glitch. So
No
I.
problem. No issue.
Okay, sir.
This zebrafish model.
Sir, you are not audible clearly. Please.
Can you hear me now? Hello. Can you hear
Yes,
me
sir. Yes.
now?
Yes, sir.
Okay.
Right. Yes.
So can you tell me about the similarity of the zebrafish brain structure with reference to humans, because you are proposing it as a alternative model to the higher species. So what do you think? What are the like similarities between zebrafish versus rodents versus humans? Can you
Yes,
comment
sir.
on that?
Sir, zebrafish is basically 99% similar to humans due to their some organelles and like bones. If we are if
No, my
like
question is regarding the brain actually. I asked about the brain.
Acha,
So
okay,
narrate
sir,
in the
sorry.
brain. So just comment
Yes,
on
sir.
that.
Sir, due to their zebrafish brain, like sorry.
Okay, no issues. So can you tell me, like, are there any specific zebrafish strains which are available? Can you name at least 2 to 3 strains of zebrafish which are available for preclinical research?
Hmm.
No idea. Okay, no issues. So just
Okay.
last questions.
Ji, sir.
What are the ethical concerns related to the zebrafish model? Are there any
Sir,
ethical, issues or requirements are there in place?
yes, sir. So ethical consideration is must before 2018. Zebrafish ethical consideration is not must, but after 2018, ethical consideration is must for.
So what are those concerns? Can you just quickly tell me one concern?
Sir, IAC approval needed for zebrafish also.
Hmm. Okay, thank
Yes.
you. So where are you working currently?
Sir, Integral University.
Okay, okay. Thank you, Rabia. Wish you all the best.
Thank you, sir.
Thank you, Rabia.
Okay, ma'am.
Kindly turn your video off. I'll request Pappu Ghosh to kindly unmute himself, turn his video on and share his screen.
Hello,
Yes, please,
am I
please.
audible?
Yeah, yeah.
Yes. Good afternoon, everyone. So, my topic for the presentation is role of amino acid in neurodegenerative disease. And from a study, it is found that in 1990, in the total population, 4% of people were suffering from neurodegenerative disease, and compared to that, in 2019, it is found that the number is doubled up to 8.2% of people are suffering from neurodegenerative disease. These are the stat. Like a neurodegenerative disease like stroke, headache, migraine, tension type headache, epilepsy, and other type like Alzheimer disease, Parkinson disease. Like these are the and these are the stat like 37.9, 17%. Like this, it has affected the total population of India. And next is introduction. Like neurodegenerative disease is mainly occurs when nervous system is damaged or neurons are damaged. And it is because of protein accumulation or dysfunction of mitochondria and oxidative stress. Because of all this, there will be dysfunction or neurodegenerative disease. And depending on neurodegenerative disease, protein um, aggregates vary in composition and location in nervous system. In Alzheimer's disease, there are senile plaques mainly made up of beta amyloids or neurofibrillary tangles, mostly made up of hyperphosphorylated tau proteins. And in Parkinson, Lewy bodies are mainly which is also proteins. And in Huntington disease, there will be aggregate of protein like polyglutamide will be aggregate, and these are the proteins which are causing this disease like Alzheimer's disease, Parkinson disease, and Huntington's disease. And next, the role of amino acid, like different type of amino acid are uh, responsible for the neurodegenerative disease. Here, some data shown that like alanine is responsible when it's increase in the brain, there will be um, Alzheimer's disease. and this positive sign is showing that the increased amount of this amino acid will cause the disease, and that minus sign is showing that decrease in the concentration of amino acid will cause the other disease. Like that, alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, beta-alanine, leucine, different type of amino acid which are responsible for different type of neurological complications. Next is amino acid and their derivative stimulate transmission of the neural signal between the individual neurons, normalize excitation and inhibition process, and also activates dormant backup neurons. Amino acid activate growth and differentiation of new nerve cell in the event of nerve cell defect caused by the cell death resulting from hypoxia and ischemia, cerebral hemorrhage, or toxic damage. They slow down the progress of apoptosis and program cell death. From a study, it is found that low protein diet result in cognitive dysfunction in aged mice, such as learning disability, inhibition, and hyperactivity behaviors, along with decreased amino acid and a consequent loss of neurotransmitter in the brain. Deficiency deficiency of amino acid may cause tau protein.
Dear participant, your time is up. Please be ready for the questions. Unmute yourself again.
Hello. Hello.
Hello. Yes, sir.
Yeah, Pappu, can you hear me? Pappu
Yes, sir.
Ghosh.
Yes, sir.
Okay, so you have presented the work on like role of amino acids in neurodegenerative disorders, right? So
Yes,
can
sir.
you name me one important protein which is responsible for the Parkinson disease?
Yes,
And
sir.
what is the like how big is this protein? Like, can you tell me about like how many amino acids are there in that? Yeah. Please go ahead.
Yes, sir. Like thyronine or tryptophan is
No. You have not got my question. I have asked you name one key protein which is responsible for the Parkinson disease, which is involved in the Parkinson disease. Is there any protein? Do you know about that?
yes, Lewy body, which is
No,
formed of
Lewy body is a different thing. I'm talking about the protein. Can you name the protein? It's alpha synuclein. Do you know how
yes,
big is
sir.
the protein?
Yes,
Okay, so can you name me some other protein which is involved in the Alzheimer disease?
like
So how it is?
amyloid beta or tau
So
protein
do you
is
know? So do you
responsible.
know how is it formed?
Like
How does amyloid beta gets formed? Any idea?
like there will be accumulation of amyloid beta.
No, I'm not asking accumulation. I'm asking how this amyloid beta proteins form inside the neurons. Do you know about the amyloid precursor protein APP?
Yes, sir.
Do you know what happened after that? Like why it is what is happening in case of patients who are suffering from Alzheimer? What is happening to these proteins APP? Because you're presenting the work on role of amino acids, so you should know about all these proteins, right, which is involved in the pathology of Alzheimer or Parkinson. These are the two key neurodegenerative disorders, right?
Yes, sir. Sir, I forgot that, sir.
Any idea about the alpha secretase or beta secretase enzymes?
Yes, sir. alpha, beta, and gamma secretes their normal condition. Alpha and gamma will secretes for
Okay. So what is the problem? Like, why this protein is, causing toxicity in case of Alzheimer disease? Why it is toxic?
like it will be uncontrollable.
No,
that-
you're not. No, that is not the case. It is an insoluble protein. Cannot,
yes,
excrete out of the system. It is keep on accumulating.
yes,
That's
yes,
why it is
yes,
formed in the plaques, right? Because it cannot
yes,
excrete out.
yes,
Okay.
sir.
So do you know which form of amyloid beta is insoluble in nature? Can you name that?
Yes. No, sir. Sorry, sir.
Okay, no issues, no issues. Thank you, Papu. Thank you for the presentation. Wish you all the best.
Thank you, sir.
Himangi, please join quickly. Pappu Ghosh, please stop sharing your screen. Are you audible, Himangi?
Yes, ma'am. Yes, ma'am.
Please start your video again. You are not visible to us.
Hello. Is the presentation visible?
Your presentation and your face both are not visible. Himangi, are you facing any difficulty?
I think we can move to the next presenter if she is not ready.
Alka Kumari, are you ready?
Yes, ma'am.
Okay, please share your screen.
Sure.
Ma'am, please turn your video on.
Yes, ma'am.
Yes, you're good to go.
Good evening, everyone. I'm here to present my topic on the role of, just a second, the role of gene alterations in immunosenescence, a review of possible novel therapeutic approach. These are the contents which I included in this slides. Introduction, alternative broads by aging immunosensing systems leading to immunosense, set of adaptive immunity and immunosense at the genetic level, and references. Introduction. Aging is among the most sophisticated and complicated biological process that may indicate the functioning of so many systems and organs, and it serves as the primary predictive marker for the functioning of genetic disorders. Immunosenescence is a combinatorial condition that has an important on both innate and acquired immunity and perform an essential aspect in chronic illness. And the word immunosenescence frequently shows the overall aging related alterations in the immune senescence. Numerous studies indicates that macrophages monocytes functions decline with aging via various mechanisms and at various stages of cell activity. This figure depicted that the role of adaptive immunity and the innate immunity, how it increases the level of inflammatory cytokines and how it gets altered the gene of aged person. Alterations brought by aging in immunosensing systems leading to immunosenescence. Cellular alterations associated with each of the form of immunity that assigned against pathogens and external materials are involved in the pathological process of immunosenescence. There are several markers which are involved in the alterations that are neutrophils, macrophages, monocytes, natural killers, or the dendritic cells. This figure depicted that immune senescence brought on by aging and defined by dysregulation of both the innate and adaptive immunosenescence. In this, these are the few cell types from the B cell and the T cells in which the senescent populations and the different markers which are involved in the alterations or change related to the cellular senescence. Immunosenescence at the genetic level. Immunosenescence tends to be most essential seen at the level of cells and molecules. Dysregulation in biological functions with aging infrequently linked to changes at the transcription level of gene. There are different factors like DNA methylation, histone, microRNA. In histone protein may be altered by a variety of ways. In addition to clinically altering DNAs, the control of helps continuing the expression of genes, and the microRNAs are also responsible for the alterations in the age-related immunosenescence. At conclusion, age-related immunosenescence system flaws diminishes the immunogenetic and the effectiveness of vaccinations in the age population. Now there aren't many researchers focusing on vaccines formulations which are essential for senior citizens, such as the Helpia B and the Fluid. But these investigations are crucial for guiding the development of novel vaccines because any immune signatures will reveal that the biological process by which these vaccines included are not good. These are the references which I included, which I referred, and that's all. Thank you.
Thank you, Alka.
So thank you, Alka, for finishing in time. So I just have
Sure.
two quick questions for you.
Yes,
Please
sir.
be brief, okay? Don't go into the details.
Yeah, sure.
Can you just name some recent discovery or any breakthrough in the field of this immunosenescence that have potential in the health or aging? Any recent breakthrough you are aware of?
No, sir.
Okay. So do you know if there is any role of epigenetics in this immunosenescence? What is the role of epigenetics?
Yes, sir, there are, but right now, exactly, I don't recall it now.
What are the different epigenetic pathways or factors? Are you aware of them?
Not now, sir.
Idea? You haven't heard about histone acetylation,
Yes, sir.
histone methylation? No idea?
Yes,
DNA
sir.
methylation?
I heard.
That's what I'm asking you. Okay,
Yes.
no problem. Thank you, Alka. Wish you all the best.
Thank you, sir.
Anshika,
Yes, ma'am.
Please turn your video on and try to share the screen. I would request Alka to please unshare the screen. Thank you, Alka. Anshika, you're visible. Your screen is also shared. Please start.
My slides are moving.
Yes, dear. Slides are moving.
Okay, ma'am. Good morning, everyone. My name is Anshika Bhatnagar from MIT University. My today's topic is 3D bioprinting in colon cancer research, a viable alternative to animal models. What is colon research? Colon cancer is a significant global health concern accounting for a large number of deaths worldwide. It is associated with chronic inflammation and increasingly with atypical inflammatory responses. This growth of cells is called as polyps. Continuous molecular subtype CMS, a classification based on differential gene expression in tumor tissue, out of which CMS2 is most common type of colon cancer. In this, the epithelial cells of colon are damaged due to cancer. WNT, wingless integration pathway, activate the MYC oncogene. There are the reasons why we need cancer research is important to us. First, for improving survival rates, reducing side effects, personalized medicine, early detection, understanding cancer biology, innovative therapies, reducing the economic burden, etc. 3D bioprinting. 3D bioprinting is an advanced technology that enables the fabrication of complex functional human tissues using bioinks composed of living cells by precisely depositing cell layer by layer. Bioprinting can create integrated tissue structures that closely mimic to the native human tissue. 3D bioprinting can be used in drug testing, disease modeling, regenerative medicine, tissue engineering, and potential for organ transplantation. Different bioprinting methods. They can be classified on the cell deposition, like first drop-based, example inkjet bioprinting, laser-assisted bioprinting. Second filament-based, example micro extrusion bioprinting. Third, a plane-based or volume-based, example stereolithography bioprinting, which is most commonly used. In this, photo decomposition is achieved through the laser beam scanning the liquid bioink. Flowchart implementation of 3D bioprinting. In this, computer-aided design is used to develop a 3D model for biofabricated structure, and bioprinting technology is used. Then bioink. Bioink are the composed of biological particles and cells. 3D design, various parameters are kept in care during this. After that, cross-linking. Cross-linking is the physical and chemical modifications that are done to that design that can be result in by enzymatic, chemical, ionic, physical, and thermal processes. At last, printing analysis, many assays like live desk assay, metabolic assays are performed to the 3D bioconstructed. at models, bioprinting allowed the fabrication of many controlled for tissue engineering spheroid organic models for oncology. Scientist named Shika Bai firstly evidenced the advantages of using CRC embedded cells in alliginated S3D in vitro models for chemotherapeutic testing, reporting on the advantage of using more relevant models to study and prevent drug resistance. In this figure, we can see from A to E, we show the HTC cells grown in biofabricated culture, and from E to H, CD44 antigen growth is shown in the early stages of cancer. Human colon cancer cell line SCT116 cells were encapsulated in alliginated beads in fabrication process to obtain 3D in vitro model. They're focusing on cell growth and expression of CD4 as a marker of stem-like cell population, tumor progression, and metastatic potential. Traditional research versus 3D bioprinting. 3D bioprinting. 3D bioprinting offers several advantages over traditional research. It follows creation of personal tissue models, which can be patient-specific cells. These models accurately represent physiochemical characteristics of individuals, enabling the targeted drug therapy. In conclusion, I would like to conclude 3D bioprinting has the potential to revolutionize the colon cancer research by providing a more ethical alternative to animal models. This technique enables the creation of realistic human tissue models, leading to the better understanding of colon cancer research.
Anshika, the time is up. Please be ready for the queries.
Yes, ma'am.
Hello.
Yes, sir.
Anshika, so okay. So do you have any idea about like kind of biomaterials, preclinical researchers using right now, or the cells, they're using in
Yes, sir.
3D bioprinting for-
Biomaterials are of two types, that are natural and synthetic. In natural, they are like protein and polysaccharides. Protein like alginate, galan gum, and so in polysaccharide like fibrin, collagen. Synthetic like PLGA, PEG.
Okay. So can you let me know what is the role of vascularization in 3D bioprinted colon cancer models?
Yes, sir. Like we have taken the HTC116 cells, that is a cell line of human colon cancer. When we encapsulate this in alliginated beads, we can have a vascularized structure of cancer cells that may represent or mimic the human colon cancer.
So you are a master student or an undergraduate student? Or
Undergraduate, sir.
undergraduate. So are you doing some research on this topic?
No, sir.
It's just the review, like kind of thing you're
Yes, sir. Like these are the references.
Mm-hmm.
These are the references.
So what do you think? What are the potential benefit of using the 3D model?
The
for the
potential
colon-
benefits, like it is of repeatability. We do not need to make models again and again. And so there are a wide range of materials available. Like the type of size we want, type of stiffness we want, we can choose material on that basis.
Okay, thank you. Thank you so much, Anshika. Wish you all the
Thank you,
best.
sir.
Thank you so much, Anshika. Afroz Jahan, please try to unmute yourself. Afroz, am I audible?
Yes, ma'am. Is my screen visible?
No, Afroz, you are also not visible. Please share your video also. You're visible. Please share your screen.
Ma'am, my screen is visible now?
No, your screen is not visible.
Ma'am, is it visible now?
No, your screen is not visible to us.
Just a second.
Do not do it in the presentation mode, otherwise it won't be visible. Afroz, are you facing any difficulty? Please inform.
Ma'am, I'm not able to share my PPT.
Afroz, can you see share screen button?
Yes,
Green
sir,
color
I'm
button.
clicking on. Yes, sir.
When you click on that button, what happens?
Sir, entire screen I have shared, but
You can
it's not.
see a menu. See menu on
Sir,
left
I have
side top
shared,
corner. You can see a screen button and click on share button.
sir, I have shared, share screen option is there. I have shared, but it is not visible. Sir, is it visible now?
No, dear.
Ma'am, please give me some time. I will reconnect.
Yeah. Afroz, you wait there. Meanwhile, Himangi, are you ready? Please try to unmute.
Yes, ma'am. Yes, ma'am.
Please try to share your screen. I can see you now.
Is it visible, ma'am? Hello? Is my screen?
Awaiting, Himangi. It's awaiting.
Okay,
It's
ma'am.
not.
It is showing your screen share is loading.
Yeah, it's visible right now. Please start.
Okay, ma'am. Good afternoon, everyone. My name is Himangivik, and my topic for today's presentation is early stage research approaches for metabolic biomarkers in Parkinson's disease. So talking about what is actually Parkinson's disease. Parkinson's disease is a adult onset neurodegenerative disorder, means where there is a degradation in the structural and functional integrity of the neurons, especially if we talk about the neurons in the nigrostriatal area, which are responsible for the production of dopamine. And Parkinson's is mainly concerned because As we can see, the pathogenesis mainly deals with the mitochondrial dysfunction, genetic mutation, oxidative disbalance, dopamine metabolism, excitotoxicity, and Levy bodies. Mitochondrial dysfunction is because some toxins alter the electron transport chain, and there is a increase in the permeability of mitochondria, which leads to the oxidative stress. Then genetic mutations, changes in the genes, oxidative disbalance, increase in the production of reactive oxygen species. Dopamine metabolism, because the neurons which are responsible for the production of dopamine, when they get destroyed in the manner of structure or function, then there is a deficiency of new dopamine. Excitotoxicity means excessive calcium deposition, and Levy bodies are the proteinaceous accumulation, which cause again the damage to the neurons. So, what are the needs to have biomarkers, or what is the requirement for the study of biomarkers? As we know, Parkinson's disease is divided into two parts: motor symptoms and non-motor symptoms. Motor symptoms include rigidity, tremor, bradykinesis, unstable posture, hypomania, etc. But non-motor functions include the pain, certain autonomic dysfunction, problem in the sleep, mood disorder. But whenever a patient start experiencing motor symptoms are evident when there is more than 50 to 70% loss in the dopaminergic neurons. But at the initial stages, their conjunctive dysfunction or autonomic dysfunction or sleep problems are the symptoms which occurs in patients. So biomarkers are needed to find out the early diagnosis of the Parkinson's disease, because the mood disturbance, conjunctive dysfunction are the common symptoms which are present in other neurodegenerative diseases also. And for particularly Parkinson's, we need to find out whether there is any mutation like alpha synuclein mutation, or there is loss of dopaminergic neurons and the level of dopamine. So that is why biomarker studies is important. Types of biomarkers for Parkinson's disease include genetic biomarkers, alpha-synuclein identification, then dopamine transport imaging, then magnetic resonance imaging, specimen-based biomarkers, gut microbiota, and inflammatory biomarkers in the blood. Because inflammatory biomarkers also, because Parkinson's is mainly caused because of inflammatory and oxidative stress also. So we will go for major three, which we will be talking about. That is, first one is patient-derived induced pluripotent stem cells, that is known as iPSCs. These are actually the somatic cells which can be reprogrammed into different types of cells that can be converted into blood cells, fibroblasts, and other cells. And now if we have reprogrammed them into different types of cells, we can easily find out, as we can see the applications. After reprogramming them, we can make them use in the disease modeling. We can create the genetic background or mutations, or we can induce the disease, and by converting them into dopamine producing neurons, we can use them for drug screening. Drug screening means we have when we go for drug development and we find out the activities, behavioral in vitro and in vivo models. So that time, with the help of these pluripotent cells, we can when we go for studies on animals, or when we go for docking studies, when we in comparison to that, if we are using these iPSCs, we can get a better result, and we can just eliminate the use of animals. Then personalized medication, as I said, it can be used in drug screening, and different atmosphere or mutations can be created using pluripotent cells. Different mutagens effect can be seen, and we can test our new medication, and we can continue with our result as we get. Next is midbrain organoids. Midbrain organoids are the three-dimensional cell cultures. The cell cultures are created by taking few cells or iPSCs from the human brain, and a model-like brain part is created.
Himangi, your time is up. Please be ready for the queries.
Yes, ma'am.
Can you hear me?
Yes, sir.
Are you an undergrad or a master student?
Sir, I'm a PhD scholar.
You're a PhD scholar in which institute?
Institute, sir, I'm a part-time PhD scholar, and currently I'm working in PSIT Kanpur as assistant professor.
Okay. Okay. So can you tell me what is the difference between biomarker and metabolic biomarker, how they are different
The
from each
metabolic biomarkers are included in biomarkers. They are one sort of biomarkers, like we can take a CSF, or we can take blood samples or urine samples and find out the metabolites in them. And we can see what the effect is having on the metabolites of the body, or what are the metabolites being generated by that particular drug.
Can you tell me, like, can you name few metabolite biomarkers, which are associated with Alzheimer or Parkinson disease?
Sir, first one is we can go for CSF testing.
No, I'm not asking the sample. I'm asking the name of the biomarker.
Sir, biomarker name. Sir, alpha synuclein test.
You think it's a metabolite biomarker?
Sir, it can be. Sir, not metabolite biomarker, but it is a biomarker.
But do you think you can measure it in patients?
No, sir, we cannot measure it quantitatively. We cannot measure it. I think so.
I'm asking if you have to diagnose the patient, you have to, understand whether this patient is prone to suffer. For example, if the patient is not suffering right now with Parkinson, if it is prone to suffer from Parkinson, so how to, utilize this, biomarker, which you're talking about, metabolite biomarker, technique to assess,
Sir, we can check for the mutations. If a person is having mutation in the genes already,
which sample you will use?
sir, we can go for genetic biomarkers like LARC mutation.
Asking about which body sample will you take?
Sir,
It's
maybe.
not possible to take the cerebrospinal fluid of patients, right?
Yes, sir.
We can, but it's very tedious, It's not that we cannot, but it's not that, patient friendly. It involves some, critical procedures. Any other sample you think you can take?
Sir, no idea.
Okay. Okay, thank you. Nice
Okay, sir.
presentation.
Thank you,
Okay,
sir.
all the best.
Thank you, sir.
Thank you, Himangi. Vishal, can you hear me?
Yes, ma'am.
Yes, please try to share your screen. Himangi, please unshare your screen. All the participants, I wanted to inform you, because of the shortage of time, we are reducing the time limit from 5 minutes to 3.5 minutes.
Is my screen visible?
So how many participants are still remaining?
Himangi, please unshare your screen.
May I know, please, how many participants are still remaining? Can you hear me? Hello?
Yes, sir.
How many participants are still pending?
Sir, give me a second.
Yeah.
Sir, we are left with more than 15 participants, sir.
Is my screen visible? Hello. Hello.
Yeah, please go ahead, Vishal. Your screen is visible and you're audible. Please go ahead.
All right. Good afternoon to one and all present over here. Myself, Vishal Gyanchandani. So today I will be talking about network pharmacology, a tool targeting reduction in the number of animals in preclinical studies. So these all are the table of the content that I will be going through. So basically, we know, first of all, this slide basically signifies the we can say the limitation of using animals in the preclinical studies. As we know that animals in the preclinical studies are basically subjected to suffering as well as debilitating pain. Also, there is a limitations of using or we can say involving the preclinical research is that it is time consuming as well as they are also cost. And at the same time, there is a question about the generalizability of the data that we are getting from the animals and extrapolation of the data from the animals to you. So considering that, there are alternative models to which researchers are looking nowadays, for example, like bioinformatics approach, to which network pharmacology belongs, then in silico methods like
QSR approach, organ on chips, microorganisms, then cell culture and tissue culturing, which are in vitro methods. So what is network pharmacology? So basically, it is an in silico technique that tries to comprehend the interaction between the bioactors and the network of the target. Here, the paradigm shift has taken from one drug, one target mode to one drug, multiple target. As we know, for example, a disease is there that is cancer, which is we can say ultimately which is responsible by the number of the genes rather than a single gene. There is an involvement of the regulatory network of the genes rather than a single gene. So cancer is a multifaceted disease. Therefore, ultimately, using one drug, one target approach is ineffective. The basic methodology of network pharmacology is data mining, network construction and analysis, and the result validation. From the data mining, we determine what are the bioactives or what are the bioactives or the drugs and the targets. In the network construction, we prepare the network and analyze it, and lastly, we do the result validation for what we have hypothesized. These all are the tools that are being used. The most widely used is Cytoscape, followed by Zephy, Pajek, and NetworkX. NetworkX basically requires the coding because it is offered by the Python. Then the next part is basically the validation. Now we will get the information about the bioactives and which are acting on the gene that is our target. Okay. So what we will be doing the analysis, that is gene ontology enrichment analysis and KEGG pathway analysis. We will see that what are the differentially expressed genes in the pathways that are contributing to the development of the specific disease. Then we will be doing the validation method. For example, like molecular docking, we will be evaluating the docking of the bioactive acting on a target, depending on the energy that we will be calculating, that is the docking score. Negative scores will define the stable configuration, followed by we will also do the microarray analysis that will be giving information about the expression of the gene, specifically log FC values. If it is negative, it is underexpressed. If it is plus, if it is positive, means if it is greater than 1, then it is overexpressed. Then there are in vitro models, like for example, the cell culture. For example, if I want to evaluate a triple negative breast cancer, so I can use MDA MB453 cell. And lastly,
Vishal, sorry, your time is up.
Vishal, I'm audible to you? Hello? Vishal, can you listen me? Please unmute. You are muted.
yes, sir.
So I just have a quick question for you. Do you think network pharmacology can replace the in vivo or in vitro experiments?
Sir, not entirely, but yes, it can mitigate, or we can say it can reduce the need of the animals in the preclinical research. Because if nowadays what happens, the research is being conducted so fast that everyone is looking for the fast results. So in order to overcome the limitations of the in vivo, we can have an alternate model, but yes, we cannot replace the animal studies.
Thank you, Vishal. I appreciate it. Thank you.
Yeah. Kanupriya, am I audible?
Yes, ma'am.
Okay, please try to share your video.
Okay.
Can you please start your video, Kanupriya?
Yes, ma'am. I'm trying to showing it, but it doesn't.
Ma'am, ma'am, is it?
Yes, yes, please start.
Good afternoon to all of you. Myself Kanupriya from Sundarvadi Pharmacy College. My topic is exploring the medicinal marvels of Berberis aristata, a comprehensive review. So today, I'm going to tell you this is my review study. So Berberis aristata is also known as Daru Haridra. It is a medicinal plant which is found in Himalaya, Nepal, and hilly areas. So present scenario of trend of herbal medicine is increased due to its lesser side effect in comparative to synthetic drug. Berberis aristata is a shrub locally known as berberry, belongs to the family of berberidae. Berberis aristata distributed from Himalaya to Sri Lanka, Bhutan, hilly area of Nepal. It grows at the height of 2000 to 3000 meter, especially in Chamba and Kumaon region of Himachal Pradesh. In Ayurveda, Berberis aristata is used as medicine for a very long time. The plant is used traditionally. The plant is used traditionally in wound healing, skin disease, inflammation, and many more other diseases. The pharmacognosy of Berberis aristata, there are many uses of this plant regarding many disease. Leaf, stems, and roots are very useful in various diseases. Propagation and cultivation of this plant for the cultivation of berberis warm moist loamy soil is required. It is shown in cold weather and it should be germinated in early season spring and or later winter. Germinating plant are transferred to the individual spots and keep in the greenhouse till first winters. Plants are growing up to 20 centimeter and then transplanted into the fields. Phytochemistry of Berberis aristata. Major phytoconstituent of Berberis are alkaloids, carotenoids, protoberberin, which is very useful in various diseases like anti-inflammatory, anti-cancers, hepatotoxicity. Bark also contain the amylin oxyberberin. These are some phytoconstituents of the Berberis aristata found in their leaves, roots, stems, and barks. Few flavonoids also present in Berberis aristata like quercetin, rutin, myridin, pectin, and there are many acids and other useful phytochemical and constituents are present. These are the some structures of the alkaloids and flavonoid which are present in the their chemical structure of the alkaloids and flavonoids present in the berberis. This is the HPLC method development of berberis. We are using HPLC method because of separation. We used to separate the chemical constituents from it. Berberin separated by HPLC on isocratic mode at 1 ml per minute. Rate of 0.1% deoxyribonucleic 60/40 ratio. Linear graph has been shown in the concentration range. HPLC is high pressure liquid chromatography, but in this we use RPHPLC, which means reverse phase high pressure liquid chromatography for its separation. These are some pharmacology of this plant used for like hepatoprotective, antioxidants, anti-diabetic, anti-hyperglycemic, these are some the part of plant like aerial fruits, roots, stems, and barks, and these are used for CCL4 induced.
Kanupriya, your time is up. Please unmute and be ready for the questions.
Madam, I don't have any questions for Kanupriya. Thank you. We can proceed with the next presenter.
Sir, she's ready for the queries.
Yeah, I
Please
said I don't have
proceed.
any specific questions for Kanupriya. Yeah, we can proceed with the next presenter quickly. Thank you. Wish you all the best, Kanupriya. Am I audible?
Yes, sir.
Yeah, wish you all the best, okay?
Thank you, sir.
Hello, Dr. Vinod. Are you facing any difficulty?
Yeah, can you hear me? Hello? Hello, can you hear me?
Yeah, sir. You're audible and visible right now. Please
Yeah,
proceed.
I think we can proceed with the next presenter. I don't have any specific questions for Kanupriya.
Okay, thank you,
Yeah,
sir.
yeah,
Okay,
please
sir.
proceed.
Vanshika, are you available? Try to unmute yourself. Suraj, are you available?
Ma'am.
Okay, please start.
Okay, ma'am.
Neeta Sinha, please be ready next.
Yes, ma'am.
Hello, ma'am.
Yes, please
Hello.
proceed.
Ma'am, my slide is visible.
Yes.
Well,
Please start.
hello. Hello. Hello, ma'am. My slide is visible.
Your slide is not visible now.
Yeah. Now, ma'am.
Yes, it's visible. Please proceed.
Okay. So, good afternoon, everyone. My name is Suraj Ramchandra Jagtap, and I am a young farm student from Novosayadri Institute of Pharmacy, Pune. I presented the review on nanoparticles and overview of preparation, characterization, evaluation, and application. Nanoparticle is a tiny science. A nanoparticle of ultrafine particles is usually defined as a particle of matter that is between 1 and 100 nanometer in diameter. Nanoparticles are considered one of the most propitious and versatile drug delivery system into the inaccessible regions like brain. It is possible or able to provide protection to therapeutic agents while sufficiently delivering them into the damaged area. So nanoparticle, we can use nanoparticle for poorly soluble drugs to improve its viability and therapeutic effects. Then advantages and disadvantages of nanoparticles. Advantages, it can be used in cosmetics and absorb much deeper into the skin. Then can be used to deliver medicines as it is readily absorbed. Then disadvantage is breathing in tiny particles could have a negative effect on your lungs. Then being used to deliver medicines nanoparticles could damage our cells as they can easily be absorbed. Then nanoparticles could accumulate in organisms over time, and we are unaware of the long-term effect of this. Then types of nanoparticles. It is divided into the two types: organic nanoparticles and inorganic nanoparticles. Inorganic nanoparticles, the polymeric nanoparticles and lipid nanoparticles. Polymeric nanoparticles are homopolymers, nanocapsules, nanospheres, copolymers, micelles, nanocapsules, polymerosomes, hydrogel nanocapsules, and dendrimers. In the lipid nanoparticles, solid lipid nanoparticles, liposomes, micelles. And in inorganic nanoparticles, metallic nanoparticle, fluorescence and carbonic nanotubes, ceramic nanoparticles. Then basic techniques for preparation of nanoparticles. These are the basic techniques for preparation of nanoparticles. First is ultrasonification, then supercritical fluid methods, then microemulsion based method, then double emulsion based method, then precipitation techniques, then solvent ejection techniques, then solvent evaporation method, then spray drying method, then solvent emulsification method. Solvent emulsification.
Suraj, your time is up. Sir, please proceed with the questions. Meanwhile, Neeta, please be ready.
Hello, Suraj,
Please
can you hear
unshare
me?
your screen also, Suraj.
Suraj, can you hear me? Hello. Can you please
Yes,
unmute yourself? I cannot hear you. Please unmute.
Yes, sir.
Yeah. So, okay. So I just have, like very quick suggestion for you. You are presenting, are you undergrad student or master student?
Master student.
You are a master student and you're presenting something, which is like 20 years old. So don't present something which is not novel. Nanoparticles we know like from like past more than few decades. If you're presenting something which is of review origin, not a research, from master student we're expecting research findings which you have carried out in your project or you might be carrying out in your lab. But if you don't have the data right now, at least present something which is new, which is exciting. Do you understand me, Suraj?
Yes, sir.
Don't present something which is of like no, I don't say no use, but very limited use. You are not educating people here because this is something which everybody knows at undergrad level even, right?
Right.
This is my suggestion for you. And yeah, the presentation was nice. Thank you.
Okay, thank you, sir.
Please unshare your screen. Neeta, please start sharing.
Ma'am, is my screen visible?
No, your screen is not visible.
Okay. Ma'am, 1 minute.
Jayant, are you ready? Please try sharing your screen.
Yes, ma'am.
Shrishti, please be ready next, quickly. Jayant,
Yes, ma'am. Yes, ma'am. Yes, ma'am. For ma'am.
quickly. Participants, we only have few 10-15 minutes left. Please respond quickly. Any
Yes, ma'am.
delay not acceptable.
मैम ओपन नहीं हो रहा मैम, पीडीएफ कर दो।
Afroz Jahan, please try sharing. Okay, Jen. Thank you.
Ma'am, I am audible.
Yes, please try to share.
Ma'am, there is a notification that I cannot share the screen if other participant is sharing.
Now please try again.
Neeta, please try to unshare.
Yes, ma'am.
Approach, please try again.
Yes, sir, I am trying.
Done, ma'am.
So is my screen visible?
No, Afroz. Mohammed Sufyan, please be ready next.
Okay, ma'am.
Ma'am, is my screen sharing?
Yes, yes.
Ma'am, is it visible now?
It's lagging.
Approach, we can see your Zoom screen. Please open your PPT file.
Mohammed Sufyan, please try sharing. Sorry, Afroz, we are not
Okay,
able
ma'am.
to.
Ma'am, my screen is visible.
Yes, yes, please start.
Okay. Good afternoon to all. My presentation topic is I'm Mohammed Sufyan. I'm from Integral University, Lucknow. My topic is in vivo models for and quantitative method for cutaneous wound healing assessment. So the first question is, what is cutaneous wound healing? Cutaneous wound healing refers to the natural biological process by which the body repairs damaged skin or tissue, restoring its structural and functional integrity. There are different types of cutaneous wounds. These may be surgical, incision, traumatic injuries, burns, ulcers, and more. There are 4 phases of wound healing. The first is hemostasis, in which the blood bleeding is terminated and prevention of blood loss occur. The second is inflammatory phase, and the third is proliferative, and last is remodeling phase. In the inflammatory phase, there is the activation and elaboration of immune response and that destroy the pathogen entering in the wound. And the proliferative phase involved in the formation of granulation tissue, new vascularization, and reepithelialization. And the last is remodeling phase, in which during this phase, the granulation tissue is substituted with a scar and the epidermis is formed from the immune cells, which either die by apoptosis or relocate to the dermis. The second question is why assess wound healing? So the answer is patient care. Accurate assessment ensure proper treatment and monitoring patient with difference. Research enables the development of effective wound healing strategies and therapies. The third is reducing complication. Timely assessment can help prevent complication like infection or chronic wounds. The fourth is last, quality of life. Aids in improving the quality of life for individuals with wounds. Then the in vivo models for cutaneous wound healing. So the first model is rodent models. Rodent models, mice and rats are commonly used for stridine cutaneous wound healing. There are genetic similarities to humans. They have genetic similarities to humans and relatively low cost make them popular choice. In rodent model, there are three types of wounds, which is wounds given to the mouse. The first is excisional wound, second incisional wound, and third is burn wounds. In the excisional wound, these wounds are generated by the surgical removal of the skin. And excisional wounds are considered to resemble acute clinical wounds, which require healing by second intention, that is the skin edges and not suture together. Incisional wound, this wounds model can be beneficial for.
Dear participant, your time is up. Sir, please proceed for the queries.
Do you know the pathophysiology of diabetic wound versus burn wound? What are the differences?
Sorry, sir.
What is the? Can you hear me, Sufyan?
Yes, sir. Yes, sir.
I'm asking, do you know the difference between pathophysiology of diabetic wound versus burn wound?
Sir, in diabetic wound, there is they are chronic wounds. They are known as chronic wounds in which the remodeling phase.
Also chronic in nature. Both are chronic in nature. I'm just asking.
Yes, sir.
In pathophysiology, how they are different from each other. Quickly, within.
So they are, sir, as a diabetic wound, they are very contagious and.
Look, can you tell me, like, in terms of marker, any specific marker, biomarker difference between both of them?
No, sir.
No issue, no issue. Thank you, Sufyan. That's all. Due to lack of time, we won't be able to continue discussion.
Yes, sir.
Nice presentation, anyways. Thank you.
Okay, sir. Thank you, sir.
Dear participant, please unshare your screen. Last call for Shrishti Chandalia. Can you respond, Shrishti? Nita Sinha, please try to share your screen again.
Yes, ma'am. Ma'am, is my screen visible?
Yes, please start.
Good afternoon, sir. I'm Nita Sinha, and I'm going to present potential drugs for cardiometabolic syndrome and basically cell-based assays. Cell assay is a laboratory technique which is used to measure specific cellular functions, properties, or responses. It helps us to gain insight into how cells behave under different conditions, which is essential for various scientific and medical applications. So cardiometabolic syndrome is a cluster of conditions that significantly decrease the risk of heart disease, stroke, and type 2 diabetes. It includes atherogenic dyslipidemia, dysglycemia, hypertension, obesity, and much more. The increase in adipose tissue combined with the decrease in lean body mass creates a significant risk for cardiometabolic syndrome. So here, nitrotic peptide receptors trigger the conversion of GT to CMP, which affects regulation of PPRA alpha mRNA expression, which affects fatty acid uptake in mitochondria and is involved in thermogenesis, lipolysis, and mitochondrial respiration. This causes a remarkable change in brown adipose tissue and white adipose tissue and heart. There is an increase in PPRA alpha expression leading to heightened thermogenesis, mitochondrial respiration, lipolysis, and there's also implications in weight management and energy expenditure. There's also a decrease in fibrosis in the heart. So these are the various cells assay used in cardiometabolic syndrome when I was reviewing it. First is cell viability and proliferation assay. First is BRDU incorporation assay. It measures cell proliferation by detecting the incorporation of BRDU, which is bromodeoxyuridine, into newly synthesized DNA using the S phase of the cell cycle. It's a common technique used to assess the rate of cell division. Ki67 staining, this is a cellular marker for active cell proliferation. Inflammation and immune response assays. So there is real-time PCR for inflammatory genes. Both is metabolism and signaling assays, and at lipid metabolism assays. So these include triglyceride and cholesterol assays to measure the level of lipids, providing insights into lipid metabolism and the risk of atherosclerosis. Fourth is cardiovascular assays. So cardiomyocyte action potential duration assays. These assays measure the duration of electrical signals in heart cells, which is crucial for maintaining cardiac rhythm. Fifth is cell differentiation and functional assays, which includes endothelial cell adhesion assays, vascular smooth muscle cells assays, and endothelial cell function assays, which includes tube formation and migration. This assess the ability of endothelial cells to form tubular structures and migrate, relevant to angiogenesis and tissue repairs. Six is cellular and molecular analysis assay. So first is multiplex protein analysis, which enables the simultaneous quantification of multiple proteins in a single sample, allowing for comprehensive protein profiling. These are the various applications for cell-based assays in cardiometabolic syndrome. First is mechanistic study. By using high-throughput screening platform, we can understand mechanisms underlying cardiovascular and metabolic disease. Cardiovascular disease modeling by like.
Thank you so much, Neeta.
So, Neeta, can you please unmute yourself? Neeta, can you please unmute yourself?
In attendees, those who have left, kindly raise your hands.
Hello, sir.
Yeah, can you hear me, Neeta?
So your voice is quite low.
Okay, can you hear me now?
Yes, sir.
Okay, so can you tell me like one or two cell-based assay for screening of anti-Alzheimer drugs?
Anti-Alzheimer drug, cell-based assays. Tryptophan dye staining, sir. Thioflavin dye staining, thioflavin, and Congo red staining, sir.
Okay. And can you tell me in brief, like in just one line, what is optogenetics? You have put it in your slide, right? In the advancements.
Yes, sir.
What is optogenetics?
Sir, optogenetics is involved. Okay, sir. So optogenesis use lights to control and manipulate cell activity. So in the context of cardiometabolic cell assay, it helps to modulate cellular functions, leading to a deeper understanding of disease pathways and potential therapeutic targets.
Can you tell me two genes which are involved in optogenetics? Name of two genes.
Sir, PPRA alpha and PPRA gamma.
No. This is not the gene which is involved in optogenetics. So it is like controlling the gene's functions by using light, right? That is optogenetics, right? You should read about channel rhodopsin and holorhodopsin, right? Blue light and yellow light, how it regulates
Yes,
gene function. But anyway, good presentation. Very good. Thank you.
Thank you, sir.
I think we can move to the next presenter. Yeah.
With this note, sir, we are done with the presentations, and I would like to conclude the ceremony. And I'm pleased to extend my heartfelt vote of thanks before you all today to be the part of this incredible event. I want to take a moment to express my gratitude to all those who have contributed to the success of this event. First of all, our HUI ma'am, our faculty members for supporting me, MIT University, Lucknow campus for believing in me. I also want to thank our guest, Dr. Vinod Tiwari, for taking out time from his busy schedule to judge our oral presentation and to be with us. Thank you so much, sir. Your insights, knowledge, and wisdom have enriched our understanding for the topic. Last but not the least, a big thank you to all the participants, my dear students who are the driving force and inspiration behind the event. Students, let us carry the knowledge and insight gained from this event with us and apply them in our lives and work to make positive contribution to the healthcare field and society as a whole. I thank you once again, everybody, for joining to MIT University, Uttar Pradesh, Lucknow campus. Thank you, one and all. Have a nice day. A note for our participants, please be there in the meeting for few minutes. There are few instructions to be sent. Please be there.
Okay, thank you. Thank you so much, madam. So, may I leave now or do I have to stay?
Yes, sir. Thank you
Okay.
so much.
Thank you.
Sir,
Thank you so much.
kindly send us the declaration of the results.
Thank you, sir. Thank you so much.
Thank you. Thank you, madam.
हाँ ये भी शेयर कर देंगे स्टूडेंट्स participants very good afternoon there is some instructions regarding the workshop first for those who are attending offline so the participants who are coming off to attend workshop offline so they have to bring acknowledgement the receipt or acknowledgement which you have got while when you have paid the registration fees along with you that will work as a gate pass for you and we will be sharing a list of participants in the gate so the security will ask you the number so you just remember your number the serial number which is written in front of your name that should be shared with the security guard. And regarding the workshop, we will be trying to teach you about the PyMol, Autodoc, and detail about how you can get the ADME properties and the similarity search using the different tool or computational tools. So those students, you have to do some work today. So you have to bring your laptops along with you. First thing, the laptop should be fully charged and you have to download some softwares in you so that the time we should not kill much time in downloading and installing the software in the auditorium. So first you have to download the PyMol. You can get it. It is a freeware from the PyMol website. You can ask for an academic license which can be shared freely with PyMol authorities. Second, you have to download Autodoc from the Autodoc software. So you have to download the Autodoc resources as well as you have to download the Autodoc tool and the version of Autodoc tool which you have to download is Autodoc 1.5.7 which will be in the name of MGL tools 1.5.7. Then after downloading these two files, what you have to do, you have to install it in your computer. It is very easy to install it. You just double click on .exe file. The software will be installed in your computer. Third thing, what you have to do, you have to download two files from your from the search engines. First is the protein file, which can be downloaded from the RCSB website. So write down RCSB in the Google and you can download the protein file. So the protein file which I am instructing is 5. You can write down the name of the protein file. It is a 5IVR. The protein code is 5IVR. So you can download it in a PDB format through RCSB website. Well, then you have to download the कौन बोल रहा है? Yes, am I audible, students?
Right.
You can type it on chat box.
प्रोटीन का नाम टाइप करो.
Yes, okay. So I am audible to everyone more or less. Thank you. Thank you, students. So the protein ID is 5IVR. The protein ID is 5IVR. And the name of the ligand which you have to download is a rincophylline. You can go to PubChem compound. You will just write down in the Google PubChem. So the PubChem web page will be open. So just write down rincophylline on the PubChem compound search, and the first compound rincophylline will be there. So download that compound in a 3D format. I am repeating that you have to download the compound in a 3D format. Okay, don't download in a 2D format because it will kill time in converting the 2D structure in a 3D structure. Okay, so these are some details which I have shared you, and rest we will be having a live sessions on the Autodoc and different tools. So you are instructed to bring your laptop fully charged. So I am repeating once more that you have to download the Autodoc resources file from the Autodoc site, and you have to download the Autodoc tool file from the Autodoc site. So two things you have to download. First, it will be Autodoc tool in the format of MGL tool, and the version you have to download is 1.5.7. That is the latest version of Autodoc, and you have to download the Autodoc resources. When you click on Autodoc resources, it will give you the options for which operating system you have to download. So you have to download for Windows, because in India, most of us have a Windows operating system. So depending upon the operating system, you can download the resources file, because without resources file, you will not be able to run the docking in the by using the Autodoc tool. And those who are attending the workshop offline, so the students, you are instructed, please bring the acknowledgement receipt or the payment receipt with you, along with you. So be on time. We will start our workshop sharp at 10. So because we have to complete it by 1 before the valedictory function, as well as before lunch. So we have shared the feedback link in the Zoom platform. So those who have not filled the feedback, please fill the feedback. And if you have any question, students, you can write down in the chat box. We are waiting. Yes, Shahzad, Hussein, Shristri, Chandelier, and Arya. What is your question? You can write down. You can write down in the chat box. Yes, so those who don't have a laptop then. So please need not to worry. Sir, you will be provided with a kit here. So that kit will contain a copy and a pen. So the instructions, the experts will run complete docking along with the analysis in the big screen. So you can go through that screen, how the person is running that docking software and how the results can be analyzed. So you can write down in your copies and you can perform it. And if you face any difficulty, you can contact it with that person. Yes, instruction for those who are attending online. So online person, the complete session will be online also. So they can watch the sessions online and they can even attend the valedictory functions online. So the complete session will be hybrid as well as online. But those who are appearing offline for workshop, they will be benefited more. Those who have Autodoc version 1.5.6, will it be okay? No, Arya, please try to download the latest version, because many bugs which are there in Autodoc version 1.5.6 has been removed. And it is in the latest version 1.5.7 that were bugs which are creating problems in protein preparation and ligand preparation, they are not there. So it is recommended that download the latest version, Autodoc 1.5.7. Okay, we will write the instructions in the WhatsApp, and please do it. Don't waste your time in downloading and installing software in the auditorium. Yes, HOI Madam, Dr. Anuradha Madam. Can we close this session for today? Yes, important instruction, as I told you, those who are attending the session offline, the student, please remember the serial number which we will share in your WhatsApp group. A list will be shared in your WhatsApp group. Please remember the serial number and your name, because huge number of participants are turning on for offline workshop. Approximately 400 candidates are listed that are attending the offline workshop. So please remember the serial number, because in the security gate, if there will be a rush, there will be unnecessary panic will be created. So software, I recommended that you download the PyMol academic version. The license is free for academic version. You download Autodoc 1.5.7 and the Autodoc resources file. The same can be posted in the WhatsApp group. I can handle 2 or more questions. If you have any more question, you can write down in the chat box. बोल दिया फीडबैक लिंक तो पोस्ट किया ओके आप कर सकते हो मैं भी ये करूँगा. Yes, by what time we have to there at the campus? So the workshop will start sharp at 10 a.m. So it is advisable that you reach the campus by 9:30 a.m. How can I download Schrodinger for docking? No, student, the Schrodinger is not free. The Schrodinger is not free, and we will not recommend it to pick any pirated software for docking here. We are expert in Schrodinger as well as in Discovery Studio also, but right now our license are being expired, so we are not giving you training in on Schrodinger and as well as in Discovery Studio. So please, as instructed, do only those things which is instructed to you. If you have any more question, you can ask me. Students, if you have any more question, you can ask me. We can close the session by 5:15. We have 4 more minutes. Yes, you can do multiple ligand docking, but it is recommended for doing the multiple ligand docking. It will come under the high throughput screening. And if you want to perform high throughput screening, you have to use Autodoc Vena. So Autodoc Vena is slightly different from Autodoc 4.2 and Autodoc NGL tool 1.5.7. So we will be dealing in single ligand docking and multiple chain docking. Okay, we will share the instruction in group as well. Shivani Pandey. Any more questions? Okay, I am sharing the feedback link once again in the group. Please give me a few minutes. So it was a wonderful day. We have a lot of presentations that belongs to the review section as well as research section. See, the students who don't attend just offline conference need to come with their PC. If you have your laptop, you can bring it with, along with you, because it is recommended that if you do the thing with your hands, you will learn more. And if you just watch it, you will forget soon. Workshop. Actually, in workshop, what we do, we will give training on certain softwares. Shivani Pandey has asked that I have never attended any conference. Can you brief know what happens in workshop? See, in a workshop, we will give you hands-on training that how you can run a software and how you can analyze the result, what you have done by using that particular software. So I have shared the feedback link. Sir, ये share हुआ क्या? Share नहीं हो रहा है. हो गया? हो गया. Okay, students, now thank you very much for attending today's international conference. We will hope we will meet in the day 3, and you will be gaining a good knowledge about different computational tools, which will be helpful in your research. Thank you very much. Sir, please close this meeting.
Yes, sir. Okay, sir.
Please
Okay.
close this meeting.
Okay,
The meeting. Okay, thank you, students. Thank you very much.